Today's topic of discussion is translation or protein biosynthesis. Translation in eukaryotics or protein biosynthesis. What is meant by translation? How to define? Then what are the requirement for this biosynthesis process? mRNA, tRNA, ribosome, amino acid, factors, it having various steps used for initiation, elongation and termination. We want to discuss. So how to define that translation? It is a process, okay, it is a process in which codon, this one is the codon, it is a process in which codon, that means genetic information in the form of codon. So these are the genetic information. Suppose this one is the mRNA, these are the things is the codon, codon in the form of codon, triplet nucleotide base pair sequence. So these are the codon genetic information in the form of codon triplet nucleotide base pair sequence based on the mRNA 5' 3' direction. So 5' or 3' direction, this codon are decoded into amino acid. This codon are decoded into amino acid for the synthesis of peptide or protein. So the process by which the information in the form of codon present on mRNA, it is used for their synthesis or translated into amino acid for the synthesis of peptide or protein. This is translation. So what is translation? This one is the mRNA 5' 3' direction. This one is the codon. Codons are decoded into amino acid. For the synthesis of peptide or protein, it is the process by which that information in the form of codon, presented on mRNA, it is used for synthesis or translated into amino acid for the synthesis of peptide or protein. This is translation. So next, what is the requirement of translation? What are the things required for translation? mRNA is required which containing genetic information or codon. These are the triplet base pair sequence. So triplet base pair sequence present on mRNA molecules read from 5' to 3' direction, non-overlapping, less unambiguous. This one is non-overlapping, not other way to interpret it. Specific, there are universal, and they follow the degeneracy, not other way into interrupt. Okay, so specific, they are universal and they follow degeneracy. What is mean by degeneracy? One means one amino acid is coded by more than one codon. One amino acid, degeneracy means one amino acid coded, okay, is coded by more than one codon. That is known as degeneracy of genetic code. So mRNA is required. So these are the mRNA, these are the things required. So the tRNA is the adapter molecules. Okay, the tRNA is the adapter molecules. It is required to carry the amino acid to the ribosome. So it required to carry amino acid to the ribosome for the synthesis of peptide or protein. So tRNA is required here. Then ribosome. Ribosome in eukaryotes. It is 8S ribosome. It is 8S ribosome. So this is it having two common subunit. Okay, this 8S, it is composed of two subunit, that is 60X and 40S. So it is composed of 60S ribosomal unit and 40S ribosomal unit. This 60S ribosomal unit, it is composed of RNA and protein. So it is composed of 20S rRNA, then 5.8S rRNA, then 5S rRNA, and nearly about 50 proteins. So here 60S, it is composed of 20S rRNA, 5.8S rRNA, 5S rRNA, and nearly about 50 proteins. Next, 40S unit, it is composed of 18S rRNA, nearly about 34 proteins. So 40S subunit, 18S rRNA, and 34S protein, 34 protein molecules. So this is the composition of eukaryotic ribosome in 60S and 40S. So it is presented on endoplasmic reticulum. The endoplasmic reticulum bedded with ribosome. So it is known as factory for protein synthesis. So this endoplasmic reticulum bedded with ribosome. So it have that it is known as factory for protein synthesis. This ribosome is also known as factory for protein synthesis. Then amino acid, nearly about 20 amino acid are required for their protein synthesis. Nearly 20 amino acid required for their protein synthesis, and different factors are required. They are known as initiation factor, elongation factor, and termination factor. The termination factor is also known as releasing factor. So here what are the factors? Initiation, elongation, and termination. This initiation factor represent eukaryotic initiation factor. Elongation factor is represented eukaryotic elongation factor. Termination factor represent eukaryotic termination factors. So the termination factor have another name, release factor. This one is responsible for termination of protein synthesis. Okay, termination of protein synthesis. These are the various steps in protein biosynthesis. First one is activation of tRNA, also called charging of tRNA, then initiation, elongation, and termination. So various steps for protein biosynthesis. Activation of transfer RNA, also called as charging of transfer RNA, initiation, elongation, and termination. So discuss these one by one. First one is activation of transfer RNA. So tRNA is activated by the attachment of amino acid. So how it will activate? Activated by attachment of amino acid to transfer RNA, that is synthesis of amino acid tRNA. So it is a two-step process. It is a two-step process. In the first step, there is activation of amino acid. First step, there is activation of amino acid. And second step, there is attachment of amino acid. Attachment of amino acid to that tRNA. That is the synthesis of amino acid tRNA. So here a two-step process. First step, there is activation of amino acid. Second one is attachment of amino acid to that arm of the tRNA, to that arm of the tRNA. So will that activation of tRNA, the amino acid with the help of ATP, amino acid, first step, amino acid with the help of ATP. Okay, in the presence of amino acid tRNA synthetase. Okay, here enzyme amino acid tRNA synthetase, it will gives enzyme AMP and amino acid complex. So this one is the activation process. First step is that activation process, amino acid combined with ATP, presence of amino acid tRNA synthetase, it will give enzyme plus AMP plus amino acid. And now this activated amino acid, it is attached to that acceptor arm. It will attach to acceptor arm, that is CCA. This tRNA, CCA and OH hydroxy group attached to attachment of amino acid. So it have that name. So it is called as charging of transfer RNA. So here tRNA, CCA, OH hydroxy group and amino acid. So it have that name, charging of tRNA. Activation and attachment of amino acid to that arm of transfer RNA. The arm of THU, it help in the recognition of that enzyme amino acid tRNA synthetase. The THU arm help for recognition of enzyme amino acid tRNA synthetase enzyme. So it is considered as group reading step in the translation process. THU considered as group reading steps in that translation process. So second is initiation. It include first step in that initiation is activation of mRNA, formation of that ternary complex, formation of ternary complex, then formation of 43X free initiator complex, then formation of 48X initiation complex, then formation of 80X initiation complex. So these are the steps in initiation. So it is initiation process having 5 steps. First one is activation of mRNA. Second one is that formation of ternary complex. Third one is formation of 43X free initiation complex. Next 48X initiation complex. Next one is formation of 80X initiation complex. So these are the 5 important steps. See one by one. Activation of mRNA in eukaryotes. Activation with the help of different factors. So it is 5 dash cap. Okay, it is a poly A tiles, we know. Okay, it is activated with the help of factor 4F. This one activated with the help of 4F, which is composed of 4G plus 4A plus 4E. These are the factors. This 4F, this is the cap binding factors. So here cap, this 4F is the cap binding factors which form complex. So it form complex. Second one is 4F cap binding complex. 4E This one is recognition of complex. Second one is 4E. This one recognition of complex. Then 4A, this one is it remove the secondary structure of mRNA. 4A remove the secondary structure of mRNA. Then 4G, it is a part of 4F. It is a part of 4F complex. It attached with the cap. Okay, this one will attach to this cap. After the formation of cap, after that formation of cap, 4F in the cap region. Now 4F in that cap region, there is attachment of 4B and poly A binding protein, PAP, poly A protein. This result the attachment of 4F, 4B help in that attachment of this mRNA to the ribosomal unit. 4B help in recognition of AUG, while poly A binding protein it help in that requirement of 40S unit to the mRNA. So this is active mRNA. So here what will happen? 4B help in that recognition of AUG, while PAP, that poly A binding protein, it help in that requirement of 40S unit to that mRNA. So it is the active mRNA. Upstream of AUG compound, which is complementary to 18S rRNA, which act as consequences of their ACG codon. So this one acts as that recognition of that ACG codon. It is known as Kovach sequence in eukaryotes. So Kovach sequence in eukaryotics. So here what are the steps? That 4B help in recognition of AUG, while that poly A binding protein help in that requirement of 40S unit to that mRNA. Upstream of AUG component, which is complementary to that 18S rRNA, which act as consensus of that ACG codon, 18S rRNA, which act as the consensus sequence of their recognition of ACG codon. So it is known as Kovach sequence in eukaryotic. In prokaryotic, it have that name Shine Dalcaro sequence. In eukaryotic, it have that name Kovach sequence. Okay, next one is formation of ternary complex. So what is ternary complex? It is formed by tRNA, eukaryotic initiation factor 2 and GTP. So this tRNA is the methionine tRNA, then eukaryotic initiation factor 2 along with GTP. This complex is known as ternary complex. Next step, 43S free initiation complex. So this is 80S ribosomal unit. It is dissociated into 60S and 40S. This 40S with the help of enzyme, with the help of eukaryotic initiation factor 6. This 40S unit initiation factor 3 and initiation factor 1A. This initiation So this initiation factor 1A prevent the reassociation of 40S and 60S into 80S. Then there is attachment. Okay, so after that there is attachment of ternary complex. So this is the factor 3, then factor 1A, then eukaryotic initiation factor 2 along with the GTP. This one is the GTP. Then this one is methionine transfer RNA. This complex, this ternary complex attached here. So here what are the things is presented? This factor 3, then factor 1A, then GTP along with that eukaryotic initiation factor 2 and this one methionine tRNA. This complex is known as 43S free initiation complex. So here that formation of 43X free initiation complex. Now this 43S free initiation complex is converted into, how it will convert 48X initiation complex. How it will convert? So here this is the factor, here same things, factor 3 presented, or that factor 1A is presented, then that methionine, this transfer RNA, tRNA is presented, GTP presented along with that 4F complex, this 4F complex, then poly A binding protein. Here this mRNA also. So factor 4B, there is AUG. So this is the methionine transfer RNA. There is, this one is the anticodon. This one is the AUG. This one is the anticodon. So corresponding region of transfer RNA in anticodon. So this complex is known as 40S initiation complex. Then with the help of factor that is eukaryotic initiation factor 5, that 60S unit will attach the ribosome. So that one will attach to that ribosome. 43X free initiation complex having factor 3, factor 1A, eukaryotic initiation factor 2 along with GTP, then methionine transfer RNA. 40S initiation complex, it containing poly A binding protein 4F, factor 3, 1A, 4B, AUG, this one is the anticodon, then methionine transfer RNA, and that anticodon, this mRNA. These are the things is presented. Now with the help of factors, that is eukaryotic initiation factor 5, eukaryotic initiation factor 5, the 60S unit will attach to that ribosome. So it will attach, 60S unit attached to that ribosome, and there is formation of 60S unit attached with 40S, 40S to form 80S initiation complex. This one is the initiation complex. So it is composed of mRNA, then tRNA. This one is the mRNA, then tRNA. This one is mRNA, this one is tRNA. So formation of ternary complex, then formation of 43S free initiation complex, 48 the S initiation complex, then 80S initiation complex. Now coming to elongation. What is elongation? It having 4 steps. Elongation, there are 4 steps. First one is attachment of amino acyl tRNA to A site. Second one is peptide bond formation or elongation of peptide chain with the help of peptidyl transferase activity. With the help of peptidyl transferase activity. Third one is translocation of ribosome. Fourth one is release of acylated tRNA. So elongation having 4 steps: attachment of amino acyl tRNA to A site, peptide bond formation, translocation of ribosome, release of acylated tRNA. So these are the 4 steps in elongation process. First one, with the help of eukaryotic elongation factor 1, or it also called as energy. Second steps, it required this one does not require any energy. There is bond formation between peptidyl tRNA, amino acid and amino acyl tRNA. First steps, it required energy. Second steps, it don't require energy. Here bond formation between peptidyl tRNA, amino acid and amino acyl tRNA. Then translocation of ribosome occur. Then next to codon. Fourth one is release of tRNA, transfer RNA from exit site. So these are the 4 basic steps in elongation. We'll discuss with this elongation. Now that new incoming tRNA, now new incoming transfer RNA with the second amino acid. First one is initiated. This one is that initiated methionine. Second one is, this one is the amino acid. Okay, second one is amino acid attached with this site. First one is methionine, second one is amino acid. So this is called attachment of tRNA A site. So this one, here what are the things presented? tRNA presented along with that peptide molecules presented. Okay, amino acid. So these two things name is called as tRNA. See the elongation. Now new incoming tRNA with this one. Second one is amino acid. First one is methionated. This one methionine. This one is the tRNA. This one is second amino acid attached with this side. Here amino acid attached with this side. So this is called as attachment of tRNA A site, whereas eukaryotic elongation factor 2 and it required energy for that attachment of A site. For that attachment of this amino acid, it required eukaryotic elongation factor 2 and energy. Next one is peptide bond formation of these 2 amino acid. So here 2 amino acid, here one more amino acid. So that peptide bond formation between 2 amino acid. Peptide bond formation between 2 amino acid. Peptide bond formation occur with the help of here with the help of peptidyl transferase activity. So with the help of peptidyl transferase activity, which is presented in that bigger unit of ribosome. This one presented in that bigger unit of ribosome. It is present in 20S, 28S rRNA. Now bigger unit of ribosome, which is presented that a bigger unit of ribosome, then it is presented in 20S rRNA. And now acylated tRNA, it is enter exit side. So this one enter to exit sites. So it is entered to exit site, then released after that translocation occur. So here translocation from A site to exit site it will move. So this is the translocation occur. Uh, this one AUG, this one is that another one is another codon, this one is one codon, this one is the another codon. Translocate, the translation of ribosome occur. So from A site to exit site it will occur. Now A site free only. So this one is translation of ribosome occur. It also required energy. Here eukaryotic factor 2 is required. For this one eukaryotic factor elongation factor 2 is required for translocation of ribosome. This translocation the ribosome move to the another position. This way transfer RNA is entered to that site. It enter peptide site. Now A site is free, so it will move to exit site. So now this A site is free. For that attachment one more new amino acid here it will come. Okay, after that free from new coming transfer RNA, so new amino acid will come. Then after that process it will reach to exit site. Now free, then one more amino acid will come. Like that only new amino acid will come, then it will reach to exit sites. This process is known as translocation of ribosome. Translocation occur. This will move to exit site, new amino acid will come. Like that only. For that attachment of new amino acid tRNA, A site free new coming tRNA. For this process is known as translocation of ribosome. These 4 steps repeated again and again until encounter that stop codon. When it will reach to stop codon, okay, up to this UAA is the will be stopped. UAA. This will stop. So next record for that attachment to that A site, then next record for the translocation of ribosome. So these are the 4 ATP record for peptide formation. So this is the termination process. So it is divided into 4 main steps. Then that is activation of transfer RNA or charging of transfer RNA, then initiation, elongation and termination. Okay, so initiation factor, elongation factor and termination process. Discuss under activation of mRNA, formation of that ternary complex, then 43X free initiation complex, 48X initiation complex, 80S initiation complex. Then elongation, it having 4 steps. What are the steps? First one is elongation. First process elongation factor 1 required. Second one is not required in energy bond formation. Third one is translocation. Then release of new transfer RNA. Then termination process required that stop codon. Okay, termination process required stop codon. After that, that one required GTP energy along with that RF factor 1 and 3. So this one is amino acid tRNA. Okay, here second step, here this one is the another amino acid. Here bond between, okay, these 2 amino acid, bond between, peptide bond formation between these 2 amino acid. 2 amino acid which attached with this site. Okay, 2 amino acid attached with this site, so it is called as tRNA A site, whereas elongation factor 1 it is required for energy for that attachment of A site. Next, what will happen? That bond formation between these 2 amino acid will occur. Bond formation between this amino acid will occur with the help of peptidyl transferase activity, which is presented in bigger unit of ribosome. Now T acetyl tRNA, this one, okay, will enter to exit site. Okay, so it will, three site is there. What are the three site? A site, B site, and exit site. So now this process occur in A site. Now it will move to E site. Now this A site free only. Now one more amino acid will come here, bonding will occur. After that it will exit to that exit site. Like that only amino acid continuously will come here, bond formation will occur. So this process will occur up to it will reach to stop codon UAA. So this one is the initiation factor, all the things. This one A side, B side, E side. Here A side, that bond formation. After that amino acid jointed with the help of peptide bond, then it will move to exit side. Now new amino acid will come. Like that only this translation process will occur.